Good afternoon, students. Today we'll start with the second chapter of biology, that is the sexual reproduction in flowering plants. So now, as you are all aware that flowering plants are the angiosperms, and the structure which is responsible for the reproductive parts is the flower. So basically we call this flower as the reproductive shoot of the flowering plants. Now a flower is a modified condensed shoot specialized to carry out sexual reproduction in higher plants. Now if we talk about a typical angiospermic flower, it consists of 4 whorls of floral appendages attached on the receptacle, that is the calyx, the corolla, the androecium and the gynoecium. Right? So of these, the 2 whorls, that is the calyx and the corolla, they are known as the non-essential parts of the flower, whereas the androecium and the gynoecium, they are the fertile and considered as the essential reproductive whorls of a flower. Now as far as these 4 whorls are concerned, we can make do with the non-essential whorls, that is if calyx and corolla is not present, then even if the reproduction will not be so much affected, because what we require mainly is the essential or the reproductive whorls, that is the androecium and the gynoecium. So in this flower diagram you can see all these different parts. Now we'll take up these one by one. You can see where petal is written, where sepal is written, right? So these are the calyx and the corolla. Now what is a sepal? Sepal is the single individual leaf segment of calyx. That is what is known as a sepal. So whorl of sepals is known as calyx, right? They are essentially green in color, but in some cases they may be colored like the petals, right? If they are colored, then such a condition of the calyx is known as petaloid. Usually they are protective in function and protect the other floral parts in bud condition because they cover it, right? So they prevent rapid transpiration from the inner parts of the flower also, so they retain the moisture. And when the flower opens, the next whorl, which is very beautiful, most of the time it is colored, different colors are there, that is known as corolla. Now where this is written, this petal, now this petal where it is written, petal is a single, you can say, segment of the corolla, right? A single individual leaf of corolla is known as a petal, and they are mostly brightly colored. Most of the brightly colored flowers are basically pollinated. It depends on the pollination. We'll take this topic a little later on, that why the flowers they have bright colors, or why they are small, why they are big. So all this depends on the type of pollination, which we'll take up a little later. Now what we now have to discuss is the next part, that is the androecium. Now androecium is the 3rd whorl of the floral appendages that arises inner to corolla. Now the individual appendage of the androecium is known as a stamen. Here you can see the androecium, and here you can see the stamen. A stamen consists of 2 parts, that is the anther and the filament, right? So if we talk about the stamen, this stamen is having the male reproductive part. We'll take this up in detail today, and the next part or the last whorl is the gynoecium or the pistil. Now this is the 4th or the last whorl, right? So they resemble a leaf. These carpels, they have floral appendages which are known as carpels. Now this gynoecium is the female reproductive organ of the plant, and it has these 3 parts, that is the stigma, the style and the ovary, Now this is the female part or the gynoecium, that is the stigma, the topmost tip, a long filament like structure, that is the style. It can be hollow or solid, and then there is a rounded bulging out structure which is known as the ovary. It has ovules inside it. Now what we'll talk about in detail today is about the male reproductive part. So when we talk about this male reproductive part, now here it consists of the anther and a long filament, right? So we'll take up the anther part first. Anther is mostly bithecous, right? It has a bilobed structure and it is bithecous. The two lobes of the diathecous anther are separated in the interior side by a deep groove and attached on the back side by a connective. Now there is a single vascular bundle in this connective. Now if we study the development of the microsporangium or of the pollen sacs, now we will see, now here is the diagram which you will see. These diagrams are very important, children, for 2 to 3 marks questions, diagrammatic type of questions. Now a very young anther of actively dividing meristematic cells, they are surrounded by a layer of epidermis, right? So it becomes two-lobed. Then each anther lobe, it develops into 2 pollen sacs. Thus a bilobed anther develops 4 pollen sacs situated at 4 corners of the anther, as you can see in the diagram. Now the development of the pollen sac begins with the differentiation of the archisporial spells in the hypodermal region, that is below the epidermis, right, at 4 corners of the young anther. You can see all this in the diagram which is illustrated over here. The archisporial cells divide by periclinal divisions to give rise to a subepidermal primary parietal layer and then an inner primary sporogenous layer. The cells of the primary parietal layer divide by successive periclinal and anticlinal divisions to form concentric layers of pollen sac wall. Now these wall layers form periphery of the from periphery to the center, they form, that is, a single layer of epidermis which becomes stretched and shrivels off when it is mature. As you can well see, I will elaborate it for you. You can see in this diagram the outermost layer, that is the epidermis layer, it will shrivel afterwards. Then a single layer of endothecium. The cells of endothecium possess fibrous thickenings. They remain thin walled and constitute stomium. Stomium is the line of dehiscence from where the pollens will come out. Mature pollens, they are shed from the line of dehiscence. In the pollens, they burst out from this point. So here the stomium part remains thin walled because it has to burst out later on. When it becomes mature and dry, the cells from here, they burst and the pollens, they are released. Then next to it is 1 to 3 layered thickness of middle layers. Cells of these layers generally disintegrate in the mature anthers, and then a single layer of tapetum. The tapetal cells, they may be uni, they may be bi or multinucleate, and they possess and cytoplasm also, and they give nourishment to the growing sporogenous tissue. So the innermost is present the sporogenous tissue. So inside, in the middle, where you see in the core of this thecus is present the sporogenous tissue. Clear? So here you see the diagrammatic layers of this diagram, and on the other side the cellular diagram is there, which shows the outer single layer epidermis, then next to it is the endothecium, then the middle layers, and then the tapetum, the nourishing cells, and innermost is the microspore mother cells. Now these microspore mother cells, they are diploid in nature. Up to here all the cells, they are having 2n number of chromosomes and they are diploid. Now the cells of this microsporangium, that is the sporogenous tissue part, it divides and then it separates from each other to function as the microsporocytes or the microspore mother cells. So the microspore mother cells, they are formed from the sporogenous tissue. So some of the microspores, they degenerate and they provide nourishment to others. The surviving microsporocytes, they are connected with each other by the cytoplasmic interconnections, that is the plasmodesmatal connections, and they have prominent diploid nuclei. The microsporocyte then develops an internal layer of callose which breaks the cytoplasmic interconnections with other microsporocytes. The separated microsporocytes then divide by meiosis and they give rise to tetrads of haploid microspores, that is by the process of cytokinesis, right? So here occurs the reductional division which is required for the formation of the gametes, that is the meiosis. So as a result of meiosis, each one microsporocyte will form 4 microspores, that is the tetrad of the microspores. And finally the cytokinesis results in the separation of these microspores. So now I am giving you some time to go through this part of the chapter, whatever we have discussed just now, and please draw these diagrams, and you can send me a photo of this diagram on the WhatsApp and I will give you the assignment on the Google Classroom, right? So thank you, children.